Good afternoon, everybody. And this is the last presentation before lunch, so we'll be as quick as possible and make it as interesting also as possible. Okay, I'm going to be talking about alternative methods for the cultivation of Cryptolepsis sanguinolenta, and this is a medicinal plant species used in the treatment of malaria. Just to give you an overview, I'll start with an introduction of the plant species itself. I'll look at previous work that was done in the domestication of this species from a grant that I received from the Volkswagen Foundation from Germany in 2011. Look at work that was entitled into the work that was done at the National Institute of Agricultural Botany under the CAPREx Fellowship. Talk about my CAPREx experience, and then I'll hand it over to Dr. Jones, who will talk briefly about the collaborations. Okay, by way of introduction, a large percentage of Africans depend on, a large percentage of our population actually depend on medicinal plants to meet their healthcare needs, populations from both the urban as well as the rural areas. And unfortunately, most of our medicinal plants, as is the case in Ghana, are collected from the wild in unsustainable ways. A case in point being Cryptolepsis sanguinolenta, which is currently on the verge of extinction. Now, just to give you a brief background, it is a perennial climbing plant, and it belongs, depending on the literature that you're looking at, it either belongs to the Apocynaceae family or the Asclepidaceae family. It has quite a number of common names, and the main one that is used in Ghana is Nibima. Now, what makes this plant very important is that it's the desired product for the plant is the root system. So here I'm showing a video of collection from the wild, and the collectors will go in with using either a hoe or cutlass, whichever one of them is going to give them as much of the plant's root system as possible. So looking at this actually makes it critical that we do something about it, because at the end of their collection, as you will see at the end of this video, very, very little of the root system is left in the ground for regeneration to take place. That's making this a critical one to look at. And interaction with the collectors, they'll tell you that they go in and whatever plant species they find, they just collect. They have also mentioned that it's becoming increasingly difficult to find these plant species in the wild. Now, look at this chemistry. Approximately 11 alkaloids have been isolated from this plant species. The main one being cryptolepin, which is the major and the most studied alkaloid found in this medicinal plant. On the local level, it's used in quite a number of antimalarials, and we did a survey, and out of 10 antimalarials that are used on the market, 7 of them actually do contain the Cryptolepsis sanguinolenta as the raw material that's used. And it was interesting to find out that most of these, they either use, Cryptolepsis is the sole active ingredient or a plant material that is used in there, or is used in combination with other antimalarials. It's also very important since one of our major alcohol producing companies, CAPREx, is actually using it in their alcoholic beverages. Now, a shift from the local market. A quick look on Amazon.com actually reveals that the plant species itself, Cryptolepsis sanguinolenta, is not only used locally in Ghana, but it's used also on the international market because of its antibacterial properties that it has. So it's quite pricey, between 50 and 70 US dollars. Now, when it comes to the conservation of this species, there are 2 ways in which you can do it. In situ, where you just leave the plant species in its environment. Unfortunately, with the advent of climate change, etc., that is not something that is feasible these days. And then there's the ex situ method, using biotechnology-based methods like tissue culture, molecular diagnostics, molecular marker technology, and cryopreservation. Now, let me just talk briefly about the research that was conducted from the Volkswagen Foundation. This started in 2011. Now, what we did with this was to quantify the active ingredient concentration in wild populations. So look at the cryptolepin levels. We looked at the relatedness amongst the populations within Ghana. We conducted quite a number of field experiments just to find out if domesticating the plants, to develop a cropping cycle. And a review of literature tells you that, once you start domesticating the plant, the chances of you losing the active ingredient becomes high. So then we conducted these field experiments to make sure that the active ingredient concentration in there is what it is that it needs to be. And then we went ahead to test the efficacy of the domesticated plants on parasitized malaria blood cell. Now, we looked at conventional plant propagation methods, and we looked at micropropagation methods, all in the bid to come up with and develop domestication protocols for this very important plant species. Now, we started with plant collections. We collected from 4 regions in Ghana: the Brongahafo, the Volta, the Ashanti, and the Eastern region. Now, what we did was, with the leaf samples, we extracted DNA, and we used that in the genetic diversity study. And then we looked at active ingredient concentration in the stem and roots of the wild population. Now, this is a graph that's just showing you location on your y-axis and amount of cryptolepin in 100 milligrams of the plant material on your x-axis. And out of the 8 different areas where collections was done, 7 out of those 8 areas, we found that the active ingredient concentration was highest in the plant's root system as compared to the stem. Now, a number of field experiments were also carried out. And in this particular field experiment, we were actually interested in if the age of the plant affected the active ingredient concentration, and if the agronomic practice of staking would also affect the active ingredient concentration. Now, this is just showing on the y-axis again, we have the amount of cryptolepin in 100 milligrams of the root material. On your y-axis, on the x-axis, sorry, we have the days after planting. And we have 0, 105, all the way to 379. Now, it was interesting to find that in the domesticated plants, at 9 months, 9 months was when the active ingredient concentration was found to be highest. And this actually coincided with the time when the average root dry weight in the plant was also highest. Now, this is important because it allows us to be able to tell the farmers who are going into its cultivation that at 9 months after planting, after sowing of your seeds, is an ideal time for you to harvest the plant. Now, we looked at propagation techniques also and quickly found that stem cuttings wasn't a viable option. So then I look at seeds, and we realized that with the seeds, you have to harvest the seeds at a mature state, just before the pods actually open up and then the seeds disperse. So in this case, you will have to bag the seeds to be able to retain them. And we found also with the seeds that the germination percentage was lost very quickly. And over a period of 1 month, you are losing the germination percentage of your seed population. Now, this led us to look at other techniques like tissue culture. And in this case, we looked at a number of experiments to determine the most effective cytokinin to use for multiplying single nodes. We looked at the ideal media for shoot induction, as well as rooting of the shoots that were developed in vitro. And look at the cropping cycle. So you start you sow your seeds in February, and it goes all the way to harvesting, which is around January to February. I mean, it's quick. I mean, we realized quickly that this seed formation is seasonal, and then also coupled with the fact that the seeds lose their viability very quickly, it shows that we need to actually look at other alternatives in terms of the conservation of this species. Now, tissue culture does offer you quite a number of advantages. It helps with germplasm conservation, and it helps with clonal multiplication of novel types. So, say we've come up with an active plant species which contains a high active ingredient concentration. It's interesting to find that tissue culture allows you to multiply that particular novel type. Now, tissue culture also gives you the advantage of generation of novel variants, and this is the focus of my CAPREx study. And in here, we're looking at callus culture and trying to generate shoots from callus and test the active ingredient in there, just to see if we could come out with some interesting types in vitro. Now, I arrived in March of this year in Cambridge, and unfortunately, the seeds that I sent ahead of time would not germinate. So I arrived and then find out that we had no seeds to work with. But what looked like a disadvantage ended up being a blessing in disguise, in that we had to look at alternatives of generating the leaf tissue that we needed for callus regeneration. So then we looked at seed sterilization protocols and developing that in vitro. We tried 6 different protocols and then came up with one. Armed with that sterilization protocol, we tried it on the 2014 seeds, as well as seeds that I brought with me in 2016. And we weren't surprised to find out germination percentages were high in seeds from 2016. But it was very interesting to find that the same seeds that would not germinate in the greenhouse were able to at least get approximately 50% germination in vitro over a 1-month period. Now, with having generated enough of the plant material, what we did was to select 10 of those plants and then collected leaf tissue from these 10 plants and then tried them on callus induction media. So we selected 7 callus induction media, 4 of them we obtained from literature, and the other 3 we got is what the National Institute of Agricultural Botany use in their callus production. So we tried those and quickly advanced and selected 4 of them, and the 4 that were selected were advanced onto shoot regeneration media. And in the process, so we had the initial material that we're working with, we had developed callus, and then we have the corresponding shoots that regenerated in vitro. So this is just to give you an idea of what went on. I mean, observations that we made. We quickly found that out of the 4 callus induction media that we were using, CS-LCM2 and CS-LCM3 ended up being better in terms of they gave us a higher number of callus induction percentage. And then the callus, the texture of the callus, it was more friable, hard, and compact, the kind that we actually needed to advance onto shoot regeneration media. We were quick to realize that W4 and LC2 gave us callus that wasn't desirable for the next phases. Now, this corresponds to, the various media that we used, and we found that if you wanted to produce a lot of callus in vitro, then the CS-LCM media was the best to do that. But if you wanted to produce callus and as well as shoot regeneration, then the best media to use was CS-LCM3 in addition to the shoot regeneration media. And we found it was amazing to find that on average you have 9 shoots per callus, having advanced it onto your shoot regeneration media. Now, not only had we been able to get the shoots to regenerate in vitro, we were also able to determine the duration that it took. So we realized that from seed to the plant, it's suitable with enough leaf tissue for callus production. It took 4 weeks. From leaf tissue to callus, another 4 weeks. And then from callus to shoot regeneration, another 4 weeks. And then from there to plantlets in vitro that are either subculture or you tend to root, it took another 4 weeks. So in a month, you have actually produced from seed all the way to regenerated shoots. Now, another interesting thing that we found was that, if you look at these were 10 plants that we took at random, and then if you look at the different plant material, we found that on the different callus induction media, what we did was to go ahead and extract, determine active ingredient concentration, which is cryptolepin, on each of the media. Now we were finding that there were a number of variations, significant variations within a particular plant species. But interestingly also that with the different plant species having been placed on CS-LCM3, you find that the coloration of your callus was actually very different. You're moving from green to a golden green and all the yellowish green, etc. So there are different variations that we found. And it's interesting to find also that this corresponded to active ingredient concentration levels within the plant. Now, this particular study is ongoing. We are almost finishing up with that. And it was interesting to find, that first of all, protocols could have been developed for the quantification of cryptolepin in vitro. Now, in summary, the CAPREx fellowship gave the opportunity for me to optimize the micropropagation protocols for the shoot multiplication for this particular plant species. Not only did we develop seed sterilization methods, but seed germination techniques also were developed. Callus induction as well as shoot regeneration media were identified for this very important plant species. We have generated at least 10 plants with their corresponding colors, as well as their regenerated shoots. And then we've been able to also quantify cryptolepin levels in the colors. So the protocol for that has also been established. Now, armed with the cropping cycle that we have, and then with dissemination materials that were developed, a video as well as video in both English and a local language, one of the local languages in Chi, and then with the production manual as well as the return on investment calculators, we are now in a better position to actually take the technology to the farmers or small scale and actually get them to stop collecting from the wild and actually go into its production. When it comes to the benefits of the CAPREx fellowship, I've benefited immensely from this fellowship, training in tissue culture techniques by the experienced NIAB crop transformation team. And I've been able, during that process also, while I was in Cambridge, expanded my research network by attending some of the conferences that were organized and joining a few of the societies around. And then my collaborator and I submitted a joint proposal to BBSRC. Although that didn't get funded, we are looking at other alternatives and other places that we could look into. And it was amazing to be able to be a part of, see firsthand, the NIAB in vitro chrysanthemum germplasm collection. And this houses the whole of Europe's chrysanthemum collection. And it was interesting to looking at that, just to forming ideas, that we could actually use with the conservation of our medicinal plant species as well. Now, it wasn't all hard work. There was time also to have fun and to do other things in Cambridge. And I'll hand it over to Dr. Jones, who will speak briefly on it.
To be very brief, NIAB is a crop science research institute on the edge of Cambridge. Many of you will have driven past it on the Huntingdon Road and not looked at it twice. And our focus is primarily UK based, but we do have an international group led by Dr. Leslie Boyd, who has a long history of working on rust diseases of cereals in southern and eastern Africa, and Tinashe Sharugwe, who's in our audience up here, who works extensively in Eastern Africa, looking to develop platforms for dissemination of knowledge and best practice to African farmers so that the science that gets developed in research institutes can actually move out into the field. Because we're a very practical, muddy boot sort of institution, we have strong links with plant breeding companies in the UK, and because of that, we run a statistical genetics for plant breeders course every year, 2 weeks intensive study, which attracts people not only from Britain and Europe, but from across the world, and we've had African students, and we've also taken the course to international venues, including Southern and Eastern Africa, where the content is tailored to the needs of the crop species that people are interested in looking at. We currently have research collaborations with institutions in India, Brazil, the Philippines, and of course we're trying to develop more links with the University of Ghana. The main role of NIAB within this collaboration has been to transfer the knowledge of tissue culture techniques to Naalamle, and hopefully that work will be developed in situ in Ghana. I have a very small role simply quantifying the active ingredient within the callus developed within the tissue culture, and Naalamle's briefly described the results, and we hope to get further work done when we take those regenerants and plant them out in our glass houses in Histon, and then take the material on to the next step. The other next step is to try and resuscitate the BBSRC grant application, where we wanted to have a two-pronged approach. One, to research into agronomic techniques that would be useful to African farmers, and particularly Ghanaian farmers. So it would be getting the best techniques but applying them in the context of Africa. And the other thrust would be to develop links or maintain links with pharmaceutical researchers, not only looking at the botanical diversity of Cryptolepsis, but looking at the compound diversity within individual accessions.
So I'll end by appreciating the Cambridge Africa program for making it possible for me to come over to Cambridge and conduct research. And then I'll say a big thank you to the Cambridge Africa Alborada Research Fund for making research funds available for conducting the research that they did. Of course, the National Institute of Agricultural Botany, and for being my host institute while I was here in Cambridge. A special thank you to Dr. Leslie Boyd, who is my mentor. Unfortunately, she's in another meeting, so she's not able to be here. And then, of course, Huw Jones, who has been working with me on quantifying the cryptolepin concentration in the callus tissue. Then, of course, to say a big thank you to the crop transformation team, head by Dr. Emma Wallington, and they have been fantastic in that their experience, they helped shape the research, shared their experience and poured out, what they knew. I mean, there were several times that we were stuck, one place or the other, brainstorming and helping us to move the project forward. And I'll say a big thank you to all of them, and thank you also for listening.